Hi. All right. So you ready? Ah, ah, ah. Let's do it. So let's do this one here. Mm. Share screen. Yep. So this one is about the continuing of this chapter 2. So this is so in chapter 2 we are talking about the organic molecule. And what's the common theme of this molecule? The common theme is that they have the monomer and they have the polymer. So we talk about carbohydrate, monosaccharides, disaccharides, polysaccharides, and we also talk about the lipid, fatty acid, uh, phospholipid, triglyceride. Yep. So it's recording, right? Yep. Let me double check. Yeah, it's recording. Good. Yeah. So the process of these molecules could be, the direction could be that breaking it down, hydrolysis, and also put it back together, dehydration synthesis. And this two direction, catabolism and anabolism combined together is the metabolism. So our body do this all the time. When we eat it, we break down the large molecule to become the monomer. So our intestine can absorb it. If there's no enzyme, then we cannot break it down. So enzyme is very important. Enzymes are proteins. So that's our next topic. We are going to talk about this molecule, protein. Protein is so important. All right, like other molecules, like other organic molecule, proteins also fit into the theme that it's polymer. Proteins are polymers. And so proteins are polymers. Then what is the monomer? The monomer of protein is amino acid. So it's right here. You can see it right here. The amino acid. Here, laser point. This right here, an amino acid. So that is the monomer. And so the monomer is the amino acid, one connect to the other, become the polymer. The two amino acid, three amino acid, four, one connect to the other. Um, they they form this chain. If it's short, it's called a peptide. If it's medium short or medium long, it's called polypeptide. It's really, really long. It will be like a string. A string usually doesn't really have the function, but that string can be folded. It's like a knitting. You have this line, this cloth. You can knit it together, make it like a cloth, a glove, something like that, and that become a functional protein. So proteins are abundant in the body, very important organ system. Uh, Not like carbohydrate and lipid. Proteins, each protein has specific function. Lipid, carbohydrates, what's their function? They are fat. They are fuel. They are like we burn it. Protein is different. Protein has its function. You won't burn your enzyme for your fuel. You are not that desperate. You have this insulin. You yourself take all the effort, have this specific mechanism, factory to produce, to manufacture this insulin as an enzyme, as a protein, and then release it. This is specific key. They are open door for what? For glucose, and you won't burn this as an energy source, right? That's too desperate. Yeah, there would be time that if you are really that desperate, that if the person is starving to the death, then you will start to burn all the protein. But before that, this key is important. We don't usually burn protein. Proteins are functional stuff. So what's their function? So their function is not just their main function is not provide the fuel. Their function is to provide very unique biochemical like a reaction. So for example, protein has these functions to support structured protein. That's this one here. I don't have a lot. Keratin, your hair is a keratin. This is a protein. And my hair is getting longer. It's because of the staying home order, social distancing. I want to write a story about that. My hair is like become like Einstein. But I haven't. I'm working on preparing my first week. But we have this discussion activity like ice breaking. Like what you do in your like staying home order. A lot of you guys, I really salute you guys that work at the hospital. I went to the hospital like yesterday to do my blood test. I have a like regular test. I don't want to talk about that, but I went to the hospital and yeah, I feel that these people, facing like patients like regular every day. I really appreciate them. All right. So what are we talking about? Proteins, right? Yeah. So that's some proteins provide that structure information, like our hair, muscle. Muscle is actually part of the movement because we can move. It's because myosin and actin actually, bind together, so they provide that movement. Transport. We just mentioned one protein actually. Just very like 2-3 slides ago, one protein conduct that transport. What's that? Apolipoprotein. That's right. Apolipoprotein is a protein to transport lipid. And what's that protein combined with lipid? What's that particle? What's the name of that particle? Lipoprotein. Lipoprotein is that name. It can be high density lipoprotein, low density lipoprotein, but that lipoprotein is not a protein. It's a particle. It's a bus carrying lipid inside of it. The driver, the carrier is apolipoprotein. That one, apolipoprotein is a protein. Lipoprotein is a particle. Buffer system, we have the major buffer system in our blood is the albumin, that is a large protein. And we have several proteins participate regulation of the acidity level in our fluid. Metabolic regulation, enzyme. So we just mentioned about enzyme to break down these disaccharides. So what enzyme is lacked if the person has lactose intolerance? That is an enzyme, right? The answer is lactase. Okay, so adults without lactase have the intolerance. Coordination, controlling hormones. So here you go, hormone. The majority of hormones are protein, but not all. We have a group of hormone that is not protein, that is lipid. It's called steroid hormone. So if you forgot, just look back to the previous talk. antibody, that's also proteins. We mentioned this, you probably heard about this like every day if you watch news, that we're supposed to, if we can detect the body's antibody to see whether the person have been affected by the COVID-19 or not. If the person has that experience, their body will build that defense antibody in the blood. So by detecting that antibody, we will know if the person has that experience or not. So this antibody is also protein. So protein has a lot of function. We don't burn it. We don't use it as fuel. We have it, we keep it intact, and that they can be modified different way, and their purpose is to provide different function. Monomer is the amino acid. So let's take a look at this monomer, amino acid. This is a typical structure of an amino acid. It has one central carbon. It has the amino group on one side. Carboxylic acid group, this very much like fatty acid, has also this part in one end. So these are all very typical. We have about 22 amino acids relevant to human physiology, and that 2022 are different in the way that they all have different R group. So this is an amino acid. An amino acid, one can link to the other, and that polymer, this is monomer, the polymer become the protein. So here is a list of this amino acid. These amino acids, the name here represent the difference in this R group. And so if you take the biochemistry before, you probably would have spent a lot of time to read, to understand all this. But here we don't need to memorize all this. We just need to know that there are so many different kinds of amino acid, and their combination will make this protein different, unique feature. So one thing you can pay attention is we have some called essential amino acid. It's highlight yellow. Can you see the yellow here? Okay, yeah. Some of these are essential fatty acid. We earlier also learned about the essential fatty acid. Here is essential amino acids. So what the essential means? What does this essential means? So essential means here, essential. When we talk about anything essential, it means that our body cannot produce it, and that we need to take it from the diet. So that's the essential means. So the question, the typical exam question related to that term is that what is defined as essential, nutrients? Is that very important to our body? It is, but it's not the answer. Is that something we can produce, we can build in the body? Is that something we cannot build, and we have to take it from the food? Yes. So that's the one. So these two amino acids can link together to form gradually, to form from the monomer to form the polymer. And then, a lot of this connection will form protein. This bonding is called a peptide bond. The process is called dehydration synthesis. This is very much like what we have learned, right? I don't really know why is this line here. This one has that too. Okay, yeah. So and the breakdown is called hydrolysis. Okay. Usually the peptide bond is very tight. It's not very easy to break down a protein. So in a way that protein is quite stable. And this process requires enzyme. So without enzyme, this connection would be very this reaction would be very difficult. So that is the monomer. Then each of these bits considered as an amino acid. Then 2 bits connect together. If it's short, it's called a peptide. If it's medium short, it's called polypeptide. If it's very, very long, it will form. It's not exactly a protein yet, but it will form a protein. So when these all connect together, they form like a string, And so in the very mature, not very immature form, this string is first, connect together, and usually it doesn't really have any function. So it's like you have like a line. You need to knead it in a way that it will form like a glove, something like that. But before that, if we just connect a line, it doesn't really provide any function. So you need to knead it. The term here is to fold it. We need to fold it. So this first structure is called primary structure. This structure is not functional. This is called a linear structure or primary structure. This is not functional. We need to fold it in order to make it. For example, insulin. Insulin is a key, right? So you need to have that string and fold it, make it very much like a key, so it can go there and open the door for glucose. So we need to fold it. So this folding is very important process for protein synthesis. It folded from, so this folding has 4 levels. First is primary structure. Linear structure is not functional. Second is that it will start to form secondary structure. This secondary structure has two forms. One is called alpha helix, so it's something like that. The other one is called beta pleated sheets. Beta pleated sheet. So alpha helix make it like a coil, right? And so this alpha helix can be folded in different way and form a pocket or something. And the beta sheets are forming like a 2D structure, for example, like that. Yeah. So it will appear very much like a sheet of paper. So we can form these protein as a sheet, like this piece of paper, as a 2D structure, right? And then we can further fold it into a tertiary structure, the 3D structure. So for example, if we have this sheet, we can further fold it. So this protein will then fold it in tertiary structure. Here we have 3D structure, and then it's like a channel, right? So the size can be defined that this channel can only allow glucose to go inside. For example, we can anchor this channel on the cell membrane, and so ion can go through it, for example, that. say this is the say this is the okay. So say this is the cell, and we can form this protein and to make it like a channel, right? So, and so which would be like a size would be not too big, not too small, but fit the glucose. Say this is glucose, right? If there's no channel, this glucose cannot enter into the cell because cell membrane is lipid. But with this channel, then glucose will be able to enter into the channel, enter into the cells. So that this channel is made by protein. It has to be folded. If the linear structure is not working, it's not going to be functional. It has to be folded. So that's tertiary structure. And then the quaternary structure. Quaternary structure means that it has several tertiary structured proteins together. So for example, we can have one protein form this channel, and then with another protein form this holding, right? So we have two pieces, and then put it together, this one become, we can use this protein to build like a cup, right? A holding and the channel or the carrier. This is like a pocket, and this part can be connected to some other part. So we can build something like that. Or for example, that our hair is one structure, like alpha helix, right? And then this alpha helix will be folded. This is just too thin, and then maybe three of them will be folded together. Then to make it, we have three string like a bonded together, wounded together to make it a protein. So this protein, the final protein can be three different chain. We call it different chain, heavy chain, light chain. If we talk about the myosin, they are coming from the different protein. But when they from different peptide, when they bind together, they complete these final products. So that's this four level of the folding structure. This is very basic, like a classification or the process. Protein, once it's produced, there are so many different, it's called post-translational modification that we can modify it. But this four structure, folded structure is the basic one. This is the major one. So we talk about the primary structure, like this one here. Then we have the secondary structure. These secondary structure could be the alpha helix or the beta pleated sheath. So it's right here, alpha helix or the beta pleated sheath. So these are the secondary structure. So question would be that the peptide folded as an alpha helix is that primary structure, secondary structure, tertiary structure, quaternary structure? The answer is the secondary structure. Same thing as the beta pleated sheath. And so here is some example about these proteins folded as either the alpha helix structure or the beta pleated sheath. These are all secondary structure. These are not like functional protein yet. We have to fold it to make it like a three-dimensional structure. So it has the structure. And so that's very important that by knowing the structure of the protein, we get some idea about the function of the protein. Okay, so knowing a structure of the protein is very important. For example, that virus, COVID-19, right? How will we know? Do you know how COVID-19 enter into the cell? They bind on an enzyme in our lung epithelium cell. This enzyme is called ACE2. So how will we know that? That's because that in the virus surface, so this is a virus, in the virus surface, it has the protein structure. And so by knowing that protein structure, we can do a lot of computer simulation, try to match that structure with the known receptor in our lung epithelium cells. And then we can match all these receptor with that structure. And then we can find the best matched one. And then we can get the idea that, okay, maybe that's how this virus attacks on our cell, because it has to find a way to land the virus into our cell in order to get absorbed by the cell. Once it's absorbed, then they can reproduce. And so the first binding is very critical. If they don't have the matched binding, they would not be able to enter into the human cells. And with either any like a modification, if this is human made, or evolution, that the virus just happen to have that protein on the surface. And that protein happens to be able to match with one of the human receptor. Then this virus become able to act on the human cells and to be entering into human cells. So that structure is important. So it's three-dimensional, has this different like hydrogen bond, covalent bonds. Doesn't really matter about this bond. Just remember that it forms this three-dimensional structure, and then they form this three-dimensional structure. It's called tertiary. The difference between tertiary and quaternary is that tertiary is from one string, one peptide. But you can know that in the world, everything is built based on multiple pieces. Like this bottle of water, it has one cap, one bottle. So it has one peptide to form this cap, the other one form this bottle, and then they together form this product. So that's quaternary structure. Here showing you some of the famous one. For example, hemoglobin. We will talk about hemoglobin a lot. The hemoglobin is a protein where? What's the function? What's the function? Function. What's the function? And where is it? So the function of the hemoglobin is to carry oxygen. Okay. So we learned that. Hemoglobin's function is to carry O2. And where is it? It is in red blood cells. Okay. So the location is inside of a cell in the blood, because I have this like a confusion from students all the time. This is this is the blood vessel. Hemoglobin is not inside of the blood. in the blood, we have these red blood cells. Hemoglobin is here inside of the red blood cells, okay, in RBC. And their function, carry oxygen, okay? So here is the hemoglobin. Hemoglobin is a tertiary structure. It has 4 side chain. So one here is a heme structure, but hemoglobin, each heme unit, each chain has 1 heme unit, and hemoglobin has 4 chain. This one is called chain, C-H-A-I. So each hemoglobin has 1, 2, 3, 4, 4 chain. Each chain, this heme, has the ion to carry O2. O2 can bind to it. So each hemoglobin can bind 4 O2. If it has a full occupation, then it binds 4 O2. So that's heme. Where is it? In the red blood cell. What's the function? To carry O2. Now, just a very quick review. We now learned about 2 carrying protein, okay? We have learned 2 carrier protein. What's that? This one, hemoglobin, globin to carry O2. And the other, what's that? A polypoprotein. Polypoprotein to carry what? To carry lipid. Okay, so now we learned 2. And a polypoprotein is here in the plasma. Okay, so these are the bus, right? Remember that? So this one is in the plasma. Plasma means the fluid in the blood. Hemoglobin is in the red blood cell, RBC. So that's very different. So hemoglobin, the structure is a tertiary structure. So here is the hemoglobin, for example, this, you see that this is a string. So every peptide, if we look into it, we will see here come with 4 different separate strings. And each string has to first have to fold it, form a little pocket like this one here. Like this is string, right? It first have to form this alpha helix, secondary structure. And then they will fold together, form like little pocket. Okay, so that's the tertiary structure. So that tertiary structure, then 4 of this tertiary structure bind together to form a quaternary structure. So that's the hemoglobin. Oxygen binds to it, O2 binds to it, and then it can be traveled in the blood. Where is it? In the red blood cells, and in the red blood cells. So that's the hemoglobin. Another quaternary structure of the protein is keratin. Okay, so keratin is like this one here. So that is first, that is 1 chain. And the alpha helix fold together with a second one. So that's the first one, the second one, the third one. 3 quaternary structure bind together to form this keratin or collagen. You know, they have a very similar structure, and that's part of the hair. So here is keratin, polypeptide chain. So 1 is 1 chain, and each keratin usually has 3 chains. Each chain is 1 individual string, folded tertiary structure. 3 of those produce this quaternary structure. They form this keratin. So that's the protein structure. So protein structure is very important in the way that we don't see this kind of importance when we talk about the carbohydrates, because when we talk about lipid, we don't really care about, for example, carbohydrate from the monosaccharide to disaccharide to polysaccharides. These polysaccharides, for example, glycogen, we don't really care about how glycogen is folded. It's a group. So if you don't remember the glycogen, we can go back and look at that structure, for example, like this one here. So this is the collagen. It has connection of different monosaccharides, glucose. We don't really care about this structure. We don't. So this one, we don't really care about how it's folded, how it's connected, because we're gonna burn it anyway. They are going to be burned. They are going to be burned to produce ATP. But protein is different. Protein structure is essential. If the folding is not correct, they cannot produce a function. And that is a lot of disease coming from, because proteins cannot be folded correctly. For example, the disease called Alzheimer's disease. Alzheimer's disease has a protein called peptide. It's a short one called amyloid beta. So that's a peptide. That's a shorter protein. And if this amyloid beta is folded correctly, it has a lot of good function, good physiological function in the nerve, in the brain. But if it's folded incorrectly, so it's really like chance by, chance by chance that we may just happen to maybe 1 million times to have incorrect folding of the amyloid beta. And then our cell, our neuron will clear it away. But when the person has a gene to have a tendency to fold it incorrectly, or if the person is getting older, then the clearance become insufficient, or the accumulation of this incorrect folded amyloid beta stay in the brain, then this incorrect folded amyloid beta will become there and they are sticky. They will bind with the other amyloid beta, then they will form this plaque and cause a lot of damage into the local neuronal area. And this plaque is the hallmark of the Alzheimer's disease. So the corrected folding is important to let the proteins to provide the function. So that's the protein. Okay, now when we have this folded protein, sometimes when we say we want to know that, so this protein basically is a polymer of amino acid. And sometimes we want to know this sequence, okay? What construct this protein. In order to really see this sequence, we need to unfold it. We need to unfold it. Then we can have the run through this detector to go through it and to build up the structure. So this unfolded process is called denaturization. So that's a term here, denaturization. 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 So that's the term. So sometimes we hit it. So we usually do this one in the lab that we want to understand, to study a protein. We hit it, we change the pH level and to unfold it. So this term is important. That, for example, the question is like the the term to describe an unfolding process of the protein. The term is called the denaturization. Now let's talk about another very important protein's function is the enzyme. So what's the function of the enzyme? The function of the enzyme is to allow the reaction to be conducted. So for example, we have the reaction A plus B to form the AB, like you are burning a piece of paper with the oxygen. For example, this piece of paper, if we can burn it and to produce CO2 and water, for example, and then, but it's not going to, this reaction will not happen unless we increase the temperature, right? So there is a barrier to make it happen. This barrier is called the, right here is called the activation energy barrier. So we need to, only when we provide the energy to overcome that barrier, then this reaction will happen. The function of the enzyme is to reduce that. So typically, say this barrier is pretty high, you need to heat it up to produce that kind of the mobility or kinetic, like energy level, then it will conduct this reaction. But by providing the enzyme, they will make this process much faster. So this is the function of the enzyme. So here is a enzyme. Enzyme is a protein. The enzyme is a protein. Okay, that is also a question you will see in a quiz or exam, that is enzyme a carbohydrate, polysaccharide, lipid, protein. The enzyme is a protein. So why is that important? It's because enzyme is very structurally related. So we have this other, molecule, they don't really care about the structure, but protein's structure is important. So this is why, it's because that when we have this protein, we'll fold it in a way like this one here. So it will generate a pocket like this one here. This one is one pocket, this one is another pocket. And then this pocket is called an active site. So for example, we have the glucose and fructose. This active site has this folding exactly match these glucose and fructose. So this enzyme can be used to break apart a sucrose, for example. So they have the active site, they have very high specificity. One enzyme work to break apart sucrose cannot break apart the lactose, for example. So enzymes have high specificity. The substrate is the one that will be conducted. For example, here, this one is a substrate. This one is a product. So for example, this is an example that this active site can bind specific to the glucose and the fructose. Then this enzyme can be act as the to break apart this sucrose, but not lactose. Sucrose fit into it, reaction, break apart, then we can break apart this sucrose into the monomer, break apart this disaccharide to the monosaccharide. So that's a protein. Then the very last one last but not least is another very important one. It's called nucleic acid. Nucleic acid. So nucleic acid is also the important organic molecule. It also has this similar theme. It has the monomer and the polymer. So let's look into that. So the monomer of the nucleic acid is the nucleotide, and the major polymer is that nucleotide. One will connect with the other, okay? And the typical one of this nucleotide polymer has two major ones. One is the DNA, the other one is the RNA. Okay, so we will learn about these two. Deoxyribonucleic acid, DNA. This is the very important genetic molecule, and RNA, ribonucleic acid, RNA. So these two are the major polymer. So if these two connect together, they will first form the RNA. And if we have two, okay, and if these two form this double strand alpha helix, then this one become a DNA. Okay. So this is the RNA, and that's DNA. So DNA, RNA are polymers. The monomer is the nucleic acid. Okay. And also that nucleic acid can be also derived into the other like very important cellular messengers such as the ATP. ATP basically is this nucleic acid, nucleotide with triphosphate. Okay, triphosphate, adenosine triphosphate. So here we have the phosphate group. If it has 3, then it becomes this ATP. It also can have a monophosphate, but folded in a way that cyclic adenosine monophosphate, CMB, is also a nucleotide. So let's look into that. So in this one, we will learn very basic concept. With this nucleic acid, we'll learn very basic concept about gene, about DNA, about RNA. And then the next one, when we talk about the cells, we will talk about how this is produced. Actually, the DNA, RNA are the foundation of protein production, protein synthesis. So we'll learn about that. So let's look at this monomer first, nucleic acid. So here is the typical nucleic acid. It has 3 parts. One is a central sugar, and then it binds with the phosphate group, and the other end binds with the nitrogenous base. So here is, for example, this one nucleic acid, and the second one here, and the third one here. So you can see that this one connect with the second one, the third one, the fourth one, and then this one form a polymer. Again, what's the process to combine them together? Dehydration synthesis. What's the process to break them apart? Hydrolysis. Okay, so it's the same applied to all these organic molecules, polymers. All right, so that's monomer connect together form the polymer. And one thing you need to notice is that this nucleic acid they are not all the same, right? Their difference is in the nitrogenous base, is right here, nitrogenous base right here. So they can be different. There are about 5 different nitrogenous base in human body. So we have 5 different nucleotide. So let's look at this one here. In DNA, this polymer DNA, let me use this one. In the DNA, okay, in the DNA, this polymer, this nitrogenous base coming from 4 different forms. We have total have 5 different kinds, but the DNA only use 4 of them. So they are the adenine, the guanine, the thymine, and cytosine. See, so we have the GCAT, 4 different types of the nucleotide. And they basically provide a coding of a gene. So for example, this chain, one of the chain can have say G, A, something like that, D, something like that. So they provide the coding, okay, of that DNA. DNA usually has, it must, okay, it's not usually, it has the double string, right? So it has a double string. Double strand, okay, double strand. If we unfolded it, we have one strand, the other strand. We have one strand, the other strand, okay? So here we may have a G, I really don't know why is that, GC, say, CCAD, something like that. And then it match with the next one, the pair one. This pairing following a principle, a pairing principle. G always pair with C, G always pair with C, A always pair with T. So if we see have GCCAD, the pair one will be CGC, right? Then A will be DT. Sorry, A. Say this A. The next one will be T. Okay, so these double strands, the nitrogen-based nucleotide are paired based on this principle. G always pair with C, A always pair with T. So if I give you a sequence of one strand, you should be able to produce the other sequence. And this will be, you will see this. If I don't quiz you, someone will, okay? So you must know this. Anyone give you one sequence, you will need to be able to produce the other sequence. And that production is by this principle, G pair with C, A pair with T. So here is the 2 strands of the DNA, their pair following this principle, A with T, G with C, okay? So that's DNA. And then the RNA also are polymer of the nucleotide. And in the RNA, in the DNA, we have the GCAT. The RNA, the nitrogenous base, are GCA and the U, so it's not T, it's a U. So basically that here we have the polymer of the RNA. RNA also one thing different is that R is ribonucleic. Their sugar is a ribose. DNA is the deoxyribose as their sugar. The RNA use ribose as their sugar. So that's also something different. The nitrogenous base are GCAU, so we may have the sequence like G, G, C, 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 U, A, for example, C, something like that. So G, okay. So we may have a sequence like that. So the difference between RNA and the DNA have this portion. One is DNA is double strands. RNA has single strand, so that's one thing different. DNA use deoxyribose as their sugar. RNA use ribose as their sugar. DNA has the bases, 4 different type of bases, GCAT. RNA has another 4. Actually, the other 3 are the same. The only difference is that they use GCAU instead of GCAT as their base. All right, so that's the RNA sequence. Here are showing you we have the 5 different nitrogenous base in a human body. They are GCAT, adenine, guanine, cytosine, and thymine. GCAT, we see them in the DNA. GCAU, uracil, is in the RNA. So here is a comparison of DNA. Here you can see that it's double strand DNA. This double strand, one strand will match with the other one. C pair with G, G pair with C, A pair with T, T pair with A. And RNA has only single strand. So this can be GCAU, UCG, or yeah, single strand. Now, RNA production. So what's this DNA? DNA is in our father and mother, right? So our mother and the father give one of its chromosome, and 2 combined together with a pair of the chromosome. They are the foundation of our gene, and so they contain this DNA. So DNA, we basically has first fertilized egg that will contains 23 pair, 20, oh, I really don't like this one here. All right, maybe try this one here. 23 pairs of chromosome. And the chromosome basically is the folded structure of the DNA. If the DNA is, folded very tightly, okay, they form this chromosome. 2 pair. Here is one pair, and then the other pair. Sorry, one of the pair. 2 together is a pair. So that is the chromosome basically contains this DNA. Okay, so this is our gene. And from the fertilized egg, we have this 23 pair. And it will be divided 23 pair. So 23 times 2, okay, 23. 23 pair. Okay, 23. Pair, so 23 times 2. And then it will be divided from the first, so 1 from the egg, the other one from the sperm, and they will two together will form this 23 pair, and then this fertilized egg will then become like a duplicated. 1 become 2, 2 become 4, and then it will coming from one single. This is one cell to a lot of the cell, millions, 100 millions of the cells to form our body, right? Our body, yeah. So we have the eye, we have the skin, we have the muscle, bone. They all coming from the same cell. So are all these cells have the same DNA? Yes, they all have the same DNA. They all come from the same fertilized egg. So DNA is there. And then how is RNA produced? So here is the RNA. How is RNA produced? So here is the cell, right? Cell has the nucleus. Nucleus has the DNA. Let's say just one DNA. Okay, one DNA. Now, RNA is produced from DNA. So here is a sequence of DNA. DNA, say this one. Okay, let's say this segment. This segment is going to be used to produce RNA. This DNA contains this string. Say A, C, C, G, T, T, okay? For example, like that. Then it will be produced, it will be used to produce RNA. This process is called transcription, but don't worry about that, we will talk about it later. Then RNA production, it will produce another sequence. This production, we need to know the principle though. A will produce GCAT, A will produce not T but U. C, it follows the principle as A to U, G to C, C to G. Okay, so A will produce G, C will match with G, C, G, G, C, T. Okay, G, C, A, T produce A. T will produce A, T will produce A. So this is new sequence. This sequence will be the RNA. So that's what we are talking right here. So RNA sequence is produced from the DNA. Okay, here we go. We need to know this term, transcription. It's a process. This process is called a transcription. Transcription, okay. And this process, following that principle, G match with C, C match G, A to U, T to A. Okay, so this is from the DNA, from the DNA code to the RNA code. Okay, so you learn that we have this very similar pairing principle. You should be able to pair two things. One is from one strand of DNA to the next strand of DNA, okay? The other is from the DNA to transcribe into the RNA. So you need to know how to, if I give you one DNA string, you should be able to produce the other string. If I give you that DNA ask you about what is the RNA, you should be able to produce that. So RNA has several functions. So DNA provide that code. And then what's the function of the RNA? RNA basically is the messenger. Here is messenger RNA is to produce protein. That process is called translation. So from RNA to protein to synthesize protein is process called translation. And but RNA is not just that kind of RNA. The majority of the RNA or the function that we really relevant to our physiology is the messenger RNA, or something we call it mRNA. mRNA. Okay. These are used to synthesize proteins through the process called translation. So here, this process is called transcription. Transcription is copy it over. Okay, so we still have nucleotide, from nucleotide to nucleotide. GCAT to GCAU, they are all very similar. So it's just transcribe, just copy over. Translation is two different language, okay? If you want to translate from one language to the other, that's totally two different language. So this is from the RNA that is the nucleic acid to protein. Protein is totally different. Protein is the polymer of amino acid. So this process is called translation. So we have that messenger RNA. This is more like most relevant RNA that we commonly talk about. We also have the ribosomal RNA, so it's a different group of RNA. We also have transfer RNA, tRNA. Actually, there are more than these three different types. We also have microRNA. So it's not mentioned here, but there are different RNA, different type of the RNA. The most relevant one would be the messenger RNA. Messenger RNA is the one to translate to protein. So how do our body, how does cell produce protein? It's all coming from the DNA, transcribe to the RNA, then translate to protein. All cells have the same set of DNA, but all cells have different function, okay? Like hair follicle can produce hair, right? But our skin, not all the skin can produce hair, right? So that's why I'm saying like very beginning that even though we may have the gene, and that gene may have the tendency to produce a certain type of protein, and that protein may cause disease, but they are not necessary to be expressed. It really depends on the condition. So their location, also the development of the cells. Okay, so that's very key information that the DNA capability of the DNA to produce RNA and the protein is called gene expression. Not all genes are always not necessary to be expressed. All right, that's right here. Gene expression. The process to synthesize protein from DNA is called gene expression. It includes these two processes: transcription from DNA to RNA, and translation from the RNA to protein. So here is the DNA transcription to RNA. So you need to know that if I give you one sequence, you need to be able to produce the other sequence. Then from RNA, the function of the RNA is to, this is one is called messenger RNA. So not all RNA do this work, but the messenger mRNA is the one to provide the code to produce protein. And this is the process called translation. So here V, H, L, T represent different amino acids. They connect together, form this peptide, fold it, will become the protein. This process, translation process, occur in the ribosome. Just know that in the cell, we have the nucleus. Nucleus contains DNA. And here, outside of it, we have the endoplasmic reticulum. It contains ribosome. Ribosome, this organelle is the one, is a location, is that location to produce, to conduct that translation. Organelle. This is organelle. Okay. So that's gene expression. So this one, I mean, how many slides do I have here? Ooh, still a lot, okay. So this one showing you the entire process. Here is the nucleus. It contains the, this one is a nucleus. It contains the DNA. DNA to produce the RNA is called transcription. GCAT to GCAU. Then this primary RNA can form the messenger RNA. Then this messenger RNA will going out of this nucleus, go into the ribosome. Then go into the ribosome. Over there, they can be translated into the protein. We just talked about this very essential steps of this gene expression. There are also a lot that, for example, here we have another process, but don't worry about this. This one, just don't worry about it. Just remember that DNA to RNA is called transcription. This transcription occurs in the nucleus. Then RNA to protein is called translation. This translation occurs in the ribosome. So here is each of these RNA. RNA has a GCAU, and then we will use this code to translate into the protein. And this happens in the ribosome. The way it does is that 3 of these RNA code, GCA, ACG, for example, like that, this code is used to match one amino acid. So this is a table, GCA match with the adenine. So this is one amino acid. Adenine will be here. ACG with the, my eye is not that good, but I think that's the cyanide, and that is right here. Okay, so we will have this amino acid linked together to form this polymer of the amino acid, and form this primary structure, fold it, then we can produce protein. What important here is that this one is called a codon. Each codon consists of 3 RNA nucleotide. So this number 3 is important. The common question would be, how many RNA nucleotides is used to form one codon? That answer will be 3, okay? So here is an example that how protein, for example, we talk about this insulin, right? The DNA from the nucleotide will flow out, RNA will flow out RNA first will go into here is the endoplasmic reticulum. And here we have this blue thing, is called ribosome. Then RNA going here. Then RNA will then be translated to protein. Then this protein will be folded. Here is the protein will be folded into and secreted packet inside of these vesicles. Then from the endoplasmic reticulum, they form these vesicles to the next stop. Here is the Golgi apparatus. So this one doesn't have the, right here, Golgi apparatus. So why do we need to do this second process? That is because that the first protein is not folded fully yet. This one is just premature produced. It's a string, it has all the material, but we need to fold it. And sometimes we need to combine 2 or 3 peptides together to form a quaternary structure. So that's folded process happens here in the Golgi apparatus. Then folded, when it's already, it will be stored in another vesicle and then stored here waiting for signal. So for example, if this is the beta cell, only when glucose level is high, then this beta cell produce this insulin then be released, and then protein will be floating in the body, acts as a key to open door for the glucose. So those are the DNA and RNA, okay? So and now we also have some other important cellular messenger or the molecule in the cell that are also the nucleic acid. The common one is the ATP, and the AMP. So they are the adenosine triphosphate. Say this is the nucleic acid. And if we have 3 phosphate, it's called ATP. This bonding, if we break it, it can produce energy. It contains very high chemical energy. So if we break it, it will release that energy for cell to use. So here is the 3 phosphate combined together. This is the adenosine triphosphate, ATP. So ATP is also a nucleic acid. And the ADP is diphosphate. AMP is monophosphate. So so that is the nucleic acid. And in the end, the very end, we will talk about some important micronutrients. These micronutrients are important in the way that our body needs it, but we don't really use it. We don't need a large amount of it. So these include the vitamin. They are essential to our cell's function. The mineral, sodium, potassium, calcium, magnesium, they are all very important. So we have 13 different essential vitamins. That means that essential means that what? Our body cannot produce, okay? That's the essential means. This enzyme, this vitamin acts as the coenzyme. So our body's cellular can have this reaction conducted inside of the cell. Vitamin deficiency is critical. Nowadays, we kind of already know this important vitamin in our body, not like 100%, but we know a lot, and we actually getting know more and more about this vitamin. And sometimes you will be surprised that actually, how can one disease like totally unrelated to a certain vitamin and turn out they are related. So like for example, autoimmune disease. Do you know any autoimmune disease? Yeah. So for example, the common one is multiple sclerosis. Okay, it's a neuron autoimmune disease. And turn out it's related to vitamin. And how do we find it? It's because that when we look into the people who has multiple sclerosis, we found them are has high like prevalence in the region that people has less sun exposure. So then we realized that okay, sun exposure can affect people to produce vitamin D. And then we realized, oh, maybe D is essential to human autoimmune system. And then we learned a lot about how vitamin D actually can play a role in immune system. Before that, we thought that vitamin D is for calcium. Okay, so that's vitamin D. Vitamin D, we can get vitamin D from food or from sun. Not me, but sun. Okay, sun exposure. Um, Vitamin D, traditionally, we know that vitamin D is important for calcium absorption in our intestine. So without vitamin D, even the person, so the person may have a lack of the calcium in the body. Vitamin K, we can take the vitamin K by taking the green leaf vegetable. Our body can produce some by the bacteria in our large intestine. Okay, so that is also very important. The vitamin K is important to produce these clotting factors. Later, we will learn that in our cell, we have about like 12 different kind of clotting factors produced by the liver. And the liver will use this vitamin K to produce those. So lack of vitamin K, the patient may have difficulty to form this clot. And then the patient may have a tendency to cause this vessel leakage. Here is just a table showing you all these different vitamins and their functions. And I would say that you can take a look into it, but if it's quiz, these two are essential. You only need to remember that. The question will be like, which vitamin can be acquired by sun exposure? ADKE. That's D. Which vitamin can be derived, can be gained, taken by the bacteria production in our own large intestine? That is vitamin K. Okay, so that's the only thing you really know. In addition, that we need to know that there are 4 vitamins that is lipid soluble, ADKE. Basically, AD is right here, and KE are lipid soluble. D and K and A and E. Okay, and this is a reference that you can take a look. But quiz-wise, don't really need to worry about it. Minerals, this is inorganic carbon, do not contain carbon. For example, these minerals such as calcium are important, chloride, sulfur, magnesium, phosphorus, potassium, and sodium are all very important in our body. So, and we cannot produce that. So in a way, they are essential. So here is the list of this mineral. Again, I will not question you all this because it would be too much for asking you to memorize all this. So you don't need to worry about them in the quiz or exam. We also need some antioxidants. Our body can actually produce some. So this is not entirely rely on the food or diet. But if we can take some, it's good for our health. So we need to have that idea. Typically that if you have the vegetable, like an apple or something, the color is there for a long time. That's because we have the antioxidants, that components there. Otherwise, they will be oxidized and the color will change. So those are good for you. 1 apple a day, keep doctor away. So take the vegetables. I feel like California is very fortunate area that we have like a vegetable like almost like free everywhere. You can find like almost like everyone will have a fruit tree in their house. And you don't need to really take care of it. And you have this free fruit. And even if you want to buy it, it's not that expensive. So vegetable is very good source to provide you this essential vitamin as well as these antioxidants. Here are some of the antioxidants that you can get from the diet or from the food. Again, I'm not going to quiz you about this. But you need to have that concept in order to improve your health or to maintain your health. You guys are very young, All right, that's it. Discard. All right. Email me if you have any questions, okay? Yep.